Welcome to Pharmaceutical Calculations, a Conceptual Approach, Lesson 21, Diffusion, Part 2. We have a very busy lecture today. As I mentioned in Lesson 20, diffusion is one of the most important chapters in pharmaceutical calculations. Today's objectives is to define two new parameters, permeability and flux, and then apply the Fick's law and demonstrate how to calculate the flux, the permeability coefficient, the diffusion coefficient, and proceed to describe the two pathways for passive diffusion of the drug, which is related to the absorption and distribution of the drug, and those are the paracellular and the transcellular pathways. Explain why the concentration gradient is a driving force for drug absorption. This is a very important objective here. And finally, describe the main two categories of protein transporters and their role in drug transport. And obviously, the last objective is related to the active transport, not the passive diffusion. Now, the Fick's equation, as we described in Lesson 20, is as shown here. We are going to modify this equation to incorporate two new parameters. The first one is the permeability coefficient Pm, and the second one is the mass flux. Now, the permeability coefficient is defined as the speed with which a particle diffuses through a membrane. So essentially, Pm is equal to the diffusion over the delta x, and that's why the units is, let's say, distance over time. Could be second or minute, it could be meter over minutes, and that's why it's the speed of velocity, because it's distance over time, okay? We have the units over here. Now, its magnitude depends on the characteristics of the membrane and, of course, the physical-chemical properties of the solute, so they have to match, right? For example, if you have a polar solute, its permeability will decrease with the lipophilicity of the membrane. Solute polarity, of course, and particle size. So here is the equation, the modified equation of the Fick's law that contains now the permeability. You can see you don't have delta x anymore because delta x over d over delta x is the permeability. Now you could modify it even further to include the parameter of flux, which is defined as the mass of particles that flow through a unit area per unit time. And the representative units here is, let's say, mass over the surface area over unit time. Okay, so this is the mass flux J, which is equal to 1 over A times dm dt, which is also equal to Pm to the permeability times the driving force, which is delta C. So this is the concentration on the two sides of the membrane. Okay. Now let us proceed into demonstrating how to calculate these two parameters. So here we have a real experiment actually. This is how we calculate permeability coefficients and flux in vitro. And these studies are using a cell line, CAGO2 cells, that is commonly used to determine permeability of drugs. So obviously we are growing these cells as monolayers at 37 degrees, and we are using trans wells here. And what we mean is that we grow the cells, as you can see here, as a monolayer on a porous membrane support. We have the growth media as the pink color there. Okay, so cells can grow and stay alive, right? We feed them. We add the drug on the apical side, as is shown here. That's the donor, and we receive the drug if it goes through the membrane barrier or into the basolateral side. That's the accept. Okay. Now the concentration of the drug in the donor compartment was adjusted to 0.1 millimolar. So we have added 0.1 millimolar here of drug. The cross-sectional area was adjusted to 1 centimeter square. Okay, so that's the sectional area that the drug goes through the membrane. So that's the membrane cross-sectional area. So we have basically monitor the passage of drug through that membrane barrier from donor to acceptor, and we have taken samples at these time intervals, and each time we determine the drug in basolateral side. So this column is what you see here, and this is the cumulative. So obviously 0.05 plus 0.26 is 0.31. Now, and we have to answer this question. So what is the cell apical to basolateral flux? So this is the J of the drug. What is the permeability coefficient of the drug? This is the PM. What is the diffusivity of the drug? If the thickness of the membrane monolayer is 3.4 nanometers, so this is delta X, okay, not A. A is 1 square centimeter, right? And you need to also address this question: does the drug have a potential to be administered orally based on these values, especially the permeability value, as we will see? So let's start first from the flux. Now we have our equation that the flux is equal to 1 over A times dm dt, and therefore solving our equation for dm over A, we get dm over A is equal to J times dt. Now obviously this is the time, right? And this is the y-axis. So this is the mass of drug passing through the surface area of the membrane, which is 1 square centimeter. This is the slope. This is very nice, and you can see that after 20 minutes we have a linear response here, and the slope is going to be J. That's the flux. So our slope based on the best fit equation is 0.0696, and make sure that you use the correct units here. So my slope is 0.0696 nanomoles per square centimeter per minute. Okay, so that's the flux. That's the rate of passage of the drug through that particular surface area of the membrane to the basolateral side, to the acceptor side. Okay. Now, using the same equation, we can determine the permeability coefficient. So now we can solve that equation for the permeability coefficient, and approximating delta C as being equal to C1, because the concentration of the donor, if you see the table back here again, is nanomolar compared to 0.1 millimolar. That's like almost a million times higher, this one, right? So we can safely approximate it to equal to C1. So basically delta C is equal to C1 minus C2, but this one is very close to being zero compared. I wouldn't say close to zero. I would say since C1 is much greater than C2, this one is going to be approximately equal to C1, okay? So now I can solve my equation J over C1 to determine the permeability, and this is my permeability, 0.000696 centimeter per minute. Make sure you have the right units here, okay? So this is what I have. Excuse me. The diffusion coefficient can be calculated again using the fixed equation, and now solving for the diffusion coefficient, which is PM times delta X. Delta X is given as 3.4 times 10 to the minus 7 centimeters. It was given as nanometers, and I converted it into centimeters. So now I have the diffusion coefficient of the drug is 2.37 times 10 to the minus 10 square centimeters per minute, okay? Finally, does the drug have a potential to be absorbed if it's given orally? And you need to have this piece of information. Usually high permeability drugs, in other words, drugs that have a potential to be absorbed orally, they have a permeability between 5 times 10 to the minus 6 to 10 to the minus 4 centimeter per second. So in order to be able to compare our value to these values, to this range, we have to convert the units. And when we convert the units into centimeter per second, we have 1.16 times 10 to the minus 5, which is within that range. Therefore, the drug is a high permeability drug and could exhibit a high absorption rate upon in vivo oral administration. Okay, so oral administration, in vivo oral administration. Okay, so we wrote it over here. So this is a very nice, informative, real experiment that we have cited and solved and demonstrated how we can determine the flux of the drug, the permeability coefficient, the diffusion coefficient, and eventually be able to figure out whether the drug has potential to be administered orally or we need to administer it by infusion or by injection, by an IV direct route of administration. Okay. Now let's now proceed to the passive diffusion pathways through epithelial membranes. So as I mentioned in our objectives, the drugs can passively diffuse through membrane barriers by two routes. First one is transcellular, which is the biggest route for drug absorption and distribution, and the paracellular pathway. So that's these pathways between the cells. In between the cells, as you can see, there's tight junction, and of course in between is just aqueous there. All right. So what we have here is epithelial cells, and you can see the apical side of this cell membrane is actually looking towards the lumen of the tissue or organ. The basolateral membrane domain here, okay, which is looking towards the blood side. And here is a section of the plasma membrane. And of course, we're familiar with the ingredients of plasma membrane, which is mainly lipids, cholesterol, phospholipids, and membrane proteins. So they are lipidic. So plasma membrane is lipidic in nature, right? So obviously, as we understand, hydrophobic drugs should be able to go through to passively diffuse transcellularly, whereas hydrophilic drugs can only go through membrane barrier paracellularly. All right. Okay, let's first study the paracellular transport, and we're talking about passive diffusion, obviously here. Now, hydrophilic molecules, as we mentioned, can only cross the epithelial membrane paracellularly through the tight junctions, and they do that by convection. That means they go with the solvent. The solvent obviously is aqueous. They're dissolved in the aqueous biological fluids. They don't have to strip off the solvent to slide through in between the cells, right? However, this paracellular pathway is very narrow. It's because of the tight junctions there. So only very small molecules can go through with a diameter of less than 5 angstroms. And sometimes even molecules that have molecular weight much less than 200, they may not be able to go through paracellular pathway. Okay. Now, what we know from experience is that paracellular transport does not contribute significantly to drug absorption. So drug absorption is mostly through transcellular pathway, passive diffusion of drugs through the cell membranes, okay? Now, the dimension of water molecule, this specifically for water, is about 3 angstroms. So, of course, the molecular weight is 18, right? Very small molecular weight. And exhibits a significant rate of paracellular transport. Glycerol, on the other hand, although the molecular weight of it is 92, right? And that's how it is glycerol. So it's an amphipathic molecule. It doesn't, and it has dimensions about 6 angstrom. It fails to enter the paracellular pathway. Instead, it passively crosses the epithelial barrier transcellularly with some difficulty, but it does go through at a slow rate. Okay. And there are some reports that glycerol is also transported actively through the plasma membrane using a protein transporter, which is, I believe, is called aquaporin. The paracellular pathway vary in different tissues, and that's a difference between the transcellular pathway, which is similar in most of the tissues. For example, the capillary endothelium is very leaky. We're talking about the capillary blood vessels here, right? And all small molecules are transported by convection paracellularly from the blood into the interstitial tissues, okay? Now let's proceed to transcellular transport. Obviously, this diffusion involves hydrophobic molecules. They cross the membrane barriers by diffusion. As we mentioned before, this is the major component of drug absorption and is similar in all tissues. And their transcellular rate of diffusion is driven by concentration gradient between the two sides of the membrane. That's why we're going to spend a significant amount of time here to study this particular statement that I wrote here to make sure that we understand what is this concentration gradient. So here is a brief description of the mechanism of transcellular transport at the molecular level, and it's not very different from dissolution. So essentially there must be a chemical interaction between the drug, the hydrophobic drug, and the plasma membrane. So initially, the hydrophobic drug, which has some affinity for water, of course, because it is in solution, it does collide with the phospholipid head groups of the plasma membrane, and it does provide energy needed to break some of the water solute and water and solute here. Okay, so here we have water solute. What we mean here, this is the water, that's the solvent. So these interactions between, now we have the solute, so this is the solute, the drug, let's say, right? And now I have drug and lipid, let's say, so I'm just going to write lipid here. This interaction must be stronger than the interaction of the drug with the water. So here is water. And because it's stronger, it provides the energy to interact with phospholipid, make some bonds, and then slowly slide deeper by diffusion within the plasma membrane bilayer. Driven by a concentration gradient on the two sides of the membrane, they reach the other side of the membrane. So again, I repeat this method. First, these drugs, being hydrophobic, they collide with the phospholipid head groups of the membrane. Because they have affinity for lipids much higher than that of water, they create bonds, attractive forces with the membrane. They slide through in the interior of the membrane, and slowly they get on the other side of the membrane because of the concentration gradient. Okay, now once in the cytosol, this is important, they will partition into the basolateral side of the cell membrane on the other side, right? So this is the cell, let's say. So they get first here, then we have the cytosol, but they have to partition into the basolateral side here and slowly leach into the interstitial tissues and then the blood. So the drug has to cross the plasma membrane twice in order to get into the blood. Okay, all right. So now we're ready to explain this concentration gradient. We definitely need to understand this driving force for absorption. So this is the equation we have for the flux, right? J is equal to the permeability coefficient times delta C. So this is the delta C. Now, so we can clearly see here that the mass of particles that flows through membrane, that's the flux, is proportional to this delta C, to the concentration of the drug on the two sides of the membrane. Okay, so this is what we need to remember. This is a constant, obviously. So here in this schematic, I spent some time to draw this so we can understand exactly how the delta C or the concentration difference is so important, and it is the one that drives the process of drug transport. So assuming here we have a portion of the intestine, okay, so this is the upper part, C1, that's the upper, and the bottom part of the intestine is C2, the lower, that's C2 here, okay. The thickness of the membrane is delta X, and what we see here is that since J is the flux is equal to PM times C1 minus C2, and since here we need to understand that in this case, in the first case, for the upper portion of the membrane, J is going to be equal to PM times C1 minus C blood. Okay, and for the lower part of the membrane, J is going to be equal to PM C2 minus C blood. Since C1, you see how much drag we have here, right? It's much higher than C2. The flux, or the rate of transport of the drug through the membrane, is going to be much higher than at the lower portion of the intestine. Okay, simple as that. So again, I repeat, since C1 is much higher than C blood, essentially actually the concentration of the blood, of the drug in the blood, is going to be close to zero, because the moment it is diffused in the blood is carrying it away into other tissues, right? So since C1 is much higher than C2, as you can see over here, the J, I'm just going to call it J1 here, it's going to be much higher than J2. So that's the flux of the drug. Okay, so hopefully everybody gets the picture. The higher the concentration difference between on the two sides of the membrane, the higher is the flux. And of course, in this case, in the two different portions of the intestine, since C1 is much higher than C2, the rate of drug absorption from the upper part of the intestine for this particular drug is going to be much higher than on the lower part of the intestine. Okay, very important schematic over here. So now we are proceeding to the active transporters. In general, we have two main categories of active transporters, I would say transporters because some of them are not requiring energy. So we have but it's definitely not diffusion mediated. Okay, so we have two categories of transporters, protein transporters. The first one is called uptake transporters, and they belong to the SLC category. So this is, I believe it's called solute carrier transporters superfamily. And the second category are efflux transporters, and that's the ABC superfamily. So that's the ATP binding cassette superfamily. Some of the uptake transporters are organic anion, as it says there, OATP, organic anion, and organic cation. So this is for cation transporters. And of course, we have some peptide transporters. So these are uptake transporters. Okay. The efflux transporters, these are the transporters that they basically pump the drug out of the cells. That's why they are called efflux. And the uptake transporters, of course, they pump the drug, they carry the drug inside the cells. But we're going to see that you need to know exactly where these transporters are located, on which side of the membrane, the apical side of the membrane or the basolateral, because they make a difference. Okay. So the efflux transporters, some of those representative are transporters are PGP. So this is P glycoprotein, MRP, and BCRP. This is a breast cancer resistant gene. In general, so this is very important paragraph that I've written over here. What we need to remember is that the apical side of the epithelial cells is always facing the lumen of their organ, not the blood side, whereas the basolateral side is facing the blood capillaries. Okay, so if I draw, for example, a cell here, let's say this is the lumen, and that should be the apical side. Here we are. And the basolateral side, if let's say this is the blood, so that would be the basolateral side. Okay. All right. So if you have an efflux transporter here, the efflux, the drug is that passively diffused inside the cell, will bind to these transporters and it will be pumped back into the lumen. So it will never be absorbed. Okay, or it would prevent or slow down its absorption. If you had an uptake transporter on the apical side, the uptake transporter will get it inside the cell and it will facilitate its absorption toward the blood. All right, simple as that. So this is what I have described before, so you can read it at your own leisure, your own time. It's clearly written here. So it's not, again, uptake receptors or transporters are always getting drugs inside the cells, but it does depend whether they're located on the apical side or the basolateral side of the cell, of the epithelium. Okay, efflux transporters are always pumping out drugs out of the cells. Okay, and if they're located in the apical side, they will pump them back into the lumen away from the blood, so they prevent their absorption. Okay, and finally we have reached the practice exercises for you. These are very, very interesting and exciting exercises that I have over here. A10 is also very, very nice, and you need to practice your theory about what your understanding about the driving force for absorption, for drug absorption via passive diffusion. Here is a nice problem that from the slope you should be able to calculate the permeability, and then if you divide by the thickness of the membrane, you should be able to calculate the diffusion coefficients. The answers are given for your convenience. Here you are asked to determine the flux, so that would be the J, right? So if you know the permeability and the number of other parameters, you should be able to use the information given and choose one of the answers here. And finally, a permeability experiment, a real permeability experiment. And here, in this case, you can see that the ratio of the drug in the donor versus the acceptor side is different. And the goal of this experiment is to make you think of the role of transporters in addition to passive diffusion of the drug through membranes. And lastly, this is a very, very interesting exercise that you need to consider the classification of drugs based on their solubility and permeability. So this is an official classification system that is used by the FDA, and almost every drug has to be classified according to this system here. And you need to address whether the drug has a potential to be absorbed from the GI or not, and whether you would recommend administering this drug with food or without food. And in order to decide for that, you need to know whether the drug is a high permeability drug or not, and of course the solubility of drugs. So these two parameters. And again, we have reached the end of this lecture. I would like to thank you for your attention. Until next time.